action research at Helmholtz in Würzburg, Germany. I'm Neva Caliskan. I am heading the RNA Collaborative Seminar Series in Würzburg, and I'm also one of the elected directors of the RNA Society for the term 24-25. And I would like to use this opportunity for inviting everybody to send nominations for the upcoming Spotlight series. And also, if you're interested in contributing to the Spotlights, I would also be happy to hearing from you, as I'm also leading the Spotlight series together with my colleague Daniel Kiss. So today's seminar will be by 3 very talented postdocs and 1 PhD student. Anuja Kibe, Patrick Bohn, and Sahil Sharma. Before we get started, I would like to inform everybody that it's not allowed to reproduce any of the materials presented during the virtual seminar. You will be receiving the talks uploaded in the YouTube channel. And you're welcome to watch them later. So the first talk of today will be by Dr. Anuja Kibe. She recently graduated from my laboratory, working on HIV-infected cells and ribosome profiling. The second talk will be by Patrick Bohn, who is almost finalizing his PhD in the group of Professor Edmund Smith. His talk will be on RNA structure probing using native long-read sequencing techniques. And last talk will be by Sahil Sharma, Dr. Sahil Sharma, who is a postdoc in the group of Cynthia Sharma, working on RNA-guided nuclease Cas9 in its native host. So we'll have 15-minute talks followed by 5-minute question and answers. And if you have questions, please type them into the chat window, and we'll be asking them to the speakers. And if time not allows, then you will also post them via email. So let's get started with our first speaker, Anuja. Yes, we can see it.
Anuja, you're on mute.
Wow, 2 years into a pandemic. Anyway, so hi everyone, I'm Anuja, and today I would like to talk about the translational landscape of HIV-1 infected cells. Now, HIV, we all know this one. It's been around for quite some time. It is a complex retrovirus, and like most RNA viruses, is entirely dependent on the host translational machinery to produce its proteins. However, like the host mRNAs, it translates via the cap-dependent mechanism. Sometimes HIV also manipulates the host translation system and uses non-canonical events such as internal ribosome entry sites or programmed ribosomal frame shifting to translate its proteins. Now, HIV is quite well studied. There are lots of proteomic and transcriptomic data sets out there. However, little is known about the global translational landscape when in HIV-1 infected cells. What we were particularly curious about were the alternative translation events, whether there are any novel ORFs or the formation of cryptic peptides and their function. So to study this, we employed ribosome profiling. Now, ribosome profiling, most people would be aware, but those who are not, it is essentially a technique that gives us a snapshot of the actively translating ribosomes in cells at a given time point. So we can study what's happening at a global translational level. So for our study, we use SubT1, which is a T lymphocyte cell line. We infected it with the HIV GFP virus. It's important to note that we have a frameshift mutation in the envelope gene, so this will not be translated. We collected the cells at different time points and then employed ribosome profiling and RNA sequencing. Our analysis was done with PRICE, a pipeline developed by Florian. This allows for frame resolution and enables us to detect any non-canonical translation events. We also did some diasome profiling to detect ribosome collision events, which I'll talk about later. We first analyzed the global transcriptional and translational changes. If we look at the early infection time point, which is the 8 hours, we see there are more changes in the ribosec reads than compared to RNA-sec, which could mean that the initial response is translationally regulated. However, at later time points, which is 16 and 24 hours, the changes appear to be transcriptionally driven. We see an upregulation of cholesterol metabolism, stress response, and immune response, which is known for HIV. However, what was most interesting for us was that the rRNA processing and the translation processes go down. We looked at the different translational efficiencies of the genes in the affected pathways. And again, interesting to note that the ribosomal protein genes are translationally downregulated. So this means that HIV infection does induce reprogramming in the host cells and particularly downregulates translation. So we wanted to see whether, or translation related processes. So we wanted to see whether this has any impact on global translation. So for this, we did some polysome profiling analysis. When we look at the polysome profiles of mock and 24-hour infected cells, we see an enrichment of ATS, which are the monosomes, and slight reduction in the polysomes, which represent the actively translating ribosomes in cells. Now, when we look at the ratio of polysomes to monosomes, this also decreases as infection proceeds, which says that HIV actually does suppress the global translation and particularly the initiation process to some extent. We also looked at the distribution of host mRNAs over the polysome profile. Now for GAPDH, majority of the mRNA is enriched in the polysomes. That means it's actively translated in the mock cells, whereas for the infected cells, it shifts into the monosome, which also indicates that the initiation is affected. We then checked whether this happens for HIV mRNAs. No, it does not. Now, what is super cool is that the HIV isoforms are actively translated, which means they're enriched in the polysomes at both early, which is 8 hours, and late, which is 24-hour time points of infection. Now, we check whether this has any effect on the translational efficiency of host and viral genes. Now, GAPDH and actin, which represent host genes, these see a decreasing trend as infection proceeds. However, in case of GAG and POL, these are increasing as infection proceeds with a peak at around 16 hours post-infection. This means that HIV does suppress translation initiation. However, the HIV mRNA somehow evade this process. This prompted us to look at the translational landscape of HIV. To RNA-seq, we saw that the viral RNA is consistently detected. However, RPF, so the ribosome-protected fragments of ribosec, are only detected at 16 and 24-hour, the later time points. And density of the ribosome-protected fragments are highest in the 5' UTR, as well as the overlapping regions. However, through PRICE, we were able to identify the correct frame and hence the protein which was translated. This gives us the gene expression patterns of HIV. Now, for 16 hours at earlier time points, STAT and REV, which are the early regulatory proteins and help in the post-transcriptional and translation of other proteins, are expressed. VPU is also expressed at 16 and 24 hours. It helps in the release of progeny virions. Probably that's why. However, unspliced RNA, which produces GAG and the GAG-POL polyprotein, saw the largest increase from 16 hours to 24 hours, almost 10-fold. However, given that majority of our ribosome-protected fragments originate from the 5' UTR, we thought that this ribosomal stalling could indicate potential alternative initiation events. So for this, we did ribosome profiling by treating the cells with harringtonin, which is an initiation inhibitor. So this would allow us to not only detect canonical initiation sites, but also any non-canonical upstream ORFs or internal ORFs. Even in this data set, majority of our reads were from the 5' UTR. And most interestingly, most of the peaks were located at near cognate AUG codons. So these could be putative upstream ORFs. Similarly, within the HIV-1 genome, we saw 2 novel internal ORFs, one within the POL and one within the WIF coding regions, both at near cognate AUG codons. We then designed a reporter assay in which we placed mCHIRI in the same frame as the internal ORF, which means that only if the internal ORF is translated, mCHIRI would be produced. And using this design, we saw a 25% mCHIRI expression in cells, which means that the internal ORF is actively translated in cells. But what is really cool to note is that the stop codons of both of these internal ORFs are located just downstream of D2 and D3 splice site, which means that there is a potential of generating multiple peptides through different splicing events. So we looked more into that. We were using long-read nanopore sequencing, which Patrick would talk a lot about after. Sorry for the spoiler. But one question which arises from most internal ORFs starting in the middle of the canonical ORF is, how do they initiate? So what's important to note is if we see this D1A1 or D1A2 splicing, this means that majority of the GAG-POL ORF is completely deleted, which would bring the initiation codon of these internal ORFs close to the 5' UTR upstream of any canonical start codon. Thus, the potential ribosome recruitment could be in the 5' UTR. And another interesting thing which came up in our studies is that these D2 and D3 splice sites, which are located in front of the stop codons. Now, if there is translation of the IORF over the D2, D3 and A4 splice site, this would give rise to a completely undescribed reading frame, thus a novel ORF. Now, we tried to detect our peptides via mass spectrometry. Although we could not detect any peptides from the upstream ORFs or internal ORFs directly, what we could detect is this peptide, which belongs to the novel ORFs that we identified. Additionally, we could also detect one N peptide from the envelope ORF, which is not translated in our virus, so potentially arising also from this ORF and the splicing event. Now that we have some very cool initiation data, we then looked at elongation. Now, HIV is known to have this non-canonical elongation event, which is programmed ribosomal frame shifting. Now, -1 PRF indicates that the ribosome shifts back a nucleotide and starts translating in an alternative reading frame. In case of HIV, this gives rise to the GAG-POL polypeptide. Now, frame shifting is stimulated by a frame shift stimulatory region, which in case of HIV is this slippery sequence, where the ribosome stalls and shifts back a nucleotide, and also this RNA secondary structure, which causes the ribosome to stall at the slippery site. In case of HIV, this is just a very simple stem loop, which causes the HIV to frame shift around 5 to 10% in vitro. Now, in our infected cells, using codon-resolved information and looking at the ratio of the GAG and POL footprints, the frame shifting was around 12 to 17% and remained constant throughout infection. Now, we know that frame shifting involves ribosome stalling at this slippery sequence. But when we looked closer at our ribosec reads, we did not observe any such enrichment of the ribosec reads, which could point out to ribosome stalling at the slippery sequence. So we thought this could be then due to ribosome collisions, which has been shown for viruses such as TMEB. This is why we did some diasome profiling to understand the collision events. Now, looking specifically at the vicinity of the frame shift site, we could not find any particular stalling near the slippery sequence. However, there was notable accumulation of ribosomes around 70 nucleotides upstream. So the P site of this ribosome would be at this AGG codon. Now, taking a closer look at the ribosec data set again and our frame information, we do see that a lot of the ribosomes have their P site at this AGG codon upstream of the frame shift site. Now, we wondered what could be the possible reason for this stalling. Is it similar to frame shifting, where the stalling is because of this RNA structure present downstream? So to look whether there is a structural basis for the stalling phenomenon, we did some DMS probing in HIV-1 infected cells. And here we find that the HIV frame shift site is actually located within this extended structure, which is a three-way junction with a large central bulge. We then decided to see what the dynamics of the structure is and to look at the force required to unfold it. For this, we did some single molecule optical tweezer analysis. So in this case, there are just increasing and decreasing forces which are applied to the frame shift RNA. So that we can look at the unfolding as well as the refolding behavior. And through this, we identified two unfolding steps. The second step is probably the conventional frame shift site, so the stem loop. And the second step, which is at much lower forces, is probably this large complex structure unfolding. Now that we know the forces that are unfolding it, we check whether it has any effect on frame shifting. And we realize that through our dual fluorescence reporter assays, here HIRI is expressed only if the ribosome frame shifts, that if we delete the entire extended structure, leaving only the conventional stem loop, frame shifting efficiency decreases. Even if we target this structure using an antisense oligonucleotide, frame shifting decreases to similar levels. So now we know that this alternative RNA fold actually affects frame shifting efficiency, which in turn will affect GAG-Pol expression. So this brings me to the end. I think if we looked at like HIV translation through a lot of different aspects, but just to summarize it, HIV infection did affect global translation to some extent. And we particularly believe that the initiation process is affected, which the HIV mRNAs managed to evade efficiently. We did identify some novel internal ORFs, which through different splicing and translational regulation could give rise to either fusion proteins. Now, these could have novel functions, or they could control the translation or amount of the canonical ORFs. And secondly, we found this novel RNA fold near the frameshift site, which does influence the efficiency. This could be because it decreases the number of ribosomes that reach the frameshift site or slows it down to efficiently stall at the slippery sequence. And thirdly, the ribosome stacking at GAG-Pol. Now, it could be because of the structure. Another possibility is that the structure leading to the ribosome collisions causes the ribosomes to drop off or have a potential alternative frameshift event, and this could lead to a truncated GAG variant. So obviously, we do have a lot of open questions still left because science. But I really hope that our work manages to enhance our understanding of translational regulation in HIV-infected cells of both host and viral mRNAs. Our study is under revision right now, but is available as a preprint for anyone to check out. This work has obviously been done by a lot of people and a lot of people to thank. Thanks to my PhD group, the Caliskan group or the RNAs. Most of them are present here. Thanks to Redmond's group for all of their help with the nanopore sequencing and DMS probing. Thanks to Andreas's group for all of the peptidomic work. And special thanks to Florian and Stefan for all of the bioinformatic help. Thanks also to Anka, Lars, Nick, and Cezanne for all of the valuable input, and my current postdoc group, the Bizos, for all the discussions. And thanks to everyone else for listening and up for discussion.
Thank you, Anuja. So we're waiting for questions.
Okay.
As I know the work, I don't know whether I have any open questions, but
okay. I apologize for making people sleep. It's...
do you think then I can ask a question.
Yeah.
What do you think if you go back to this, why do you think there is unspliced and spliced RNAs are differentially translated?
Because each one of them gives rise to different protein products. So we have the fully spliced that would give rise to the TAT and REV, which are the early accessory proteins. Okay, I would just stop it here. And the unspliced gives rise to GAG and GAG-Pol. So depending on when they're needed during the HIV infection cycle, it is believed that HIV regulates its splicing accordingly. So,
and how are these recognized? Do you have any idea?
The splicing event.
The spliced and unspliced RNA, is there any studies on that, on the HIV?
I guess Patrick would be better to answer this, but through long read, yes. And for our polysome profiling analysis, we use specific primers that target the junctions. So,
okay. So here are the more questions just popped up, Anuja. Thank you. Peng Yao asking, do you know if ribosome collision causes mRNA decay or truncated GAG protein degradation?
Yeah, it is shown in literature that this will cause mRNA decay, but if your question is specifically for our HIV protein, that's one of our hypotheses, but we unfortunately have not proved it yet, but it is likely that it might cause this. It would be very interesting, yeah.
Now another HIV expert, Bruno Sarguil, is here saying many thanks for the nice presentation and work. That's it. So
yeah.
That was the. So he is saying many diazomes seem to be stuck on many other sites that upstream from the frameshift. Do you have any hypothesis about what they represent?
So our hypothesis right now is also that the HIV viral RNA is quite structured. So these could be just the ribosomes trying to translate all of these various structured regions. We have not particularly looked at what these could be. A disome analysis is quite complex. So if anyone has any suggestions on how to do it, that would be awesome.
Frank Slack asked, saying, very nice talk. Thanks, Frank. Your paper title mentions novel gene expression principle. Is there evidence for broader use of this mechanism?
So novel gene expression, we were particularly referring just to the frame shifting events and the ribosome stacking and stalling, which would cause it. And also the internal ORF. So yeah, there are broader things. Sorry, I guess I didn't understand the question very well.
Yeah, perhaps since the time is limited, just want to find last question. Have you tested collecting time before 8-hour infection? Yes, no question.
No, we have not, but it would be very interesting. Yes.
Frank is saying, okay, thanks. I think that was sufficient, but you might follow up with an email. And
yes.
Last question from Bruno. I would also suggest you that you type the answer in the chat, question answer, Anuja.
Yeah.
And we move on with the next speaker. Patrick Bohn, native long-read RNA structure probing to reveal RNA interaction sites. So if you unshare, let's.
Yes.
Patrick.
Yeah. Oh, sorry.
Yeah,
I
there
think
you go.
This should work. Yeah.
Great.
Okay. Yeah, I'm really grateful to be able to present here my part of my work of my PhD, which was focused on RNA structure probing. And today I'm going to present about the method we developed based on long-read sequencing, as well as an application to use this for studying RNA interactions. So I want to start very basically. A lot of you might think of RNA as this linear molecule that has these 4 different bases, and they, in terms of mRNA, they might represent a message of protein. However, this is not the only case in which RNA is used in the cell, and in particular, by it folding into particular structures, it can have meaningful functions. So, for example, this RNA is a tRNA, in particular lys3 tRNA, and it brings this really cool function of translating codons into amino acids. Now, we are more interested, though, in a different type of RNA, which, as you've heard already about, is HIV-1. And HIV-1 is a retrovirus that has a positive sense RNA genome packaged in its particles. However, when it infects a cell, this is reverse transcribed and integrates into the host cell genome, where it can then be expressed by cellular RNA polymerase machinery. Now, intriguingly, this virus somehow is able to then later detect its own RNA and package this efficiently into virus particles again. And as there have been a lot of studies, we know that this is primarily driven by its 5' UTR, which is known to fold into very intricate structures that enable it to interact specifically with lots of different proteins and RNAs. And so one example of this is this tRNA that we saw before. which can hybridize to this primer binding site and can be used as a reverse transcription primer during the infection cycle. But also we have this stem loop 1, which at the top has a dimerization initiation sequence that allows 2 genome copies to dimerize before or during packaging. And last but not least, it also contains a protein binding site for the viral gag polyprotein. And so this is all concentrated within this relatively short region, actually, making it really an interesting place to study. Now, what is really curious about it is that, as Anuja already mentioned, the HIV unspliced mRNA is actually, or can be spliced in a lot of different isoforms that can express a variety of proteins. And most interesting for us, all of these isoforms still contain the majority of this 5' UTR. So we were curious to see how the virus is able to selectively package unspliced RNA while excluding the majority of spliced RNA from being packaged, and whether that potentially might have to do with different structures of these. So what we employed was chemical probing, as you heard before. Briefly, we use a reagent that reacts with single-stranded nucleotides, but not double-stranded nucleotides, to kind of label them. Now, this technique actually has been in use for a relatively long time, and a variety of reagents have been identified. And I think the most commonly used ones are now either shape reagents or DMS, dimethyl sulfate. And I want to highlight, focus here on dimethyl sulfate. This reacts primarily with the Watson-Crick-Franklin phase of A and C residues, and so gives us information about these nucleotides. Now, what is really nice for chemical probing is that we can do this in the cell, so we can study the RNA structure in its native state and also in virion. And we can do this at endogenous expression levels of the RNA. So currently, after modification, the position of these modification needs to be read out. And this is currently performed using reverse transcription under specific conditions. And this allows modifications to be converted into mutations. So this is why it's called mutation profiling, where we then sequence the cDNA or PCR products of these cDNA to identify positions with increased mutation rates. Now, this is commonly done with Illumina sequencing. However, you might have noticed that while it has quite a few pros speaking for it, it has a limitation, especially when looking at a very complex transcriptome like the one from HIV, because not each individual read will be able to cover all potential splice sites, so we cannot uniquely map each read to a given isoform. So in contrast, we thought we would replace this step by nanopore sequencing, which can allow us then to cover with each individual read, cover all potential splice sites, and so perform isoform resolved structure probing. So in this method, we call it nano DMS map or nanopore dimethyl sulfate mutation profiling. And here's a quick workflow of this. So, and I want to mention that Anne-Sophie did the majority of this experiment of our paper, and she really did an amazing job. So so briefly with our work, so you can either transfect proviral DNA in the case of HIV, or one can also infect actually cells with virus. Then after the cells express RNA, we can perform a chemical probing in the cells or in the virion, RNA extraction, and then reverse transcription. And here we optimized the step based on a recent report from Anna Peil's lab, who developed this marathon RT reverse transcriptase to generate longer cDNA molecules. And after that, we performed PCR and nanopore sequencing. So briefly, as I mentioned, we are mostly interested in the 5' UTR. So we designed a PCR strategy that would cover this 5' UTR for all possible isoforms. However, so these reads will contain all potential splices with the exception of D4A7, for which we used 2 different reverse transcription primers to make these specific. And so after this PCR, we were able to see a single product for the unspliced target, but a mixture of products as expected for the partially and fully spliced transcripts. And so then the neat thing about nanopore is that each individual molecule gets, or DNA molecule gets read to its full length. So indeed, when we look at the length distribution of our nanopore reads, we can see that they really correspond very well to the agarose gels. So confirming that we are capturing this diversity. And each read on its own can be classified. And so speaking of this, here we used isoquant for this classification, and that worked really well. So with around 80% of reads uniquely identified. And here you can see the relative isoform abundances for unspliced, partially, and fully spliced. And this these relative abundances were also very reproducible between different replicates. But of course, this is the more straightforward part. Now the harder part is to see whether our nanopore sequencing actually generates high quality mutation profiling data. So what we next looked at was mismatch rates, in particular with increasing DMS concentration on A, C, G, and U, or now in DNA, T residues. And we could nicely see that with increasing DMS concentration, the mismatch rates for single nucleotide mismatches rise very continuously. However, the insertion and deletions, which actually makes up the majority of sequencing errors in nanopore, appear to be flat, indicating that the signal we are looking for is for DMS is actually only in this single nucleotide mismatches, for which we actually have a quite high signal to noise ratio, supporting or explaining how we are able to achieve this high quality data. And actually we also sequenced the same RNA on Illumina to ensure that this stays true, and our data quality is pretty much equal. Now that we've established this, we started with the more interesting part of looking at the DMS reactivities of different isoforms, starting with the unspliced. So here you see a heat map where each row is one sample or one isoform, and each column is an A or C residue for which we have the DMS reactivity. And for the unspliced, we were able to probe this not only in the cell, but also in the virion. And what you can really nicely see is that actually in this primer binding site, the reactivities decrease quite a lot, indicating that this RNA that was previously single-stranded is now base paired, which can easily be explained by tRNA hybridization. And similarly, we see dimerization in the stem loop 1 loop. Now, now when we look at these different spliced RNAs, we actually see a somewhat different picture. And the majority of this difference, I think, is obvious here in this lower part of SL1. So indicating that, in spliced RNAs, indeed, stem loop 1 appears to be not folded. And so that gives us a mechanistic explanation for how these RNAs are less likely to be packaged, which is that, they don't harbor the structure that we know is critical for dimerization. Now, why they don't have this, we are still researching, but we have a few theories, including that downstream sequences might affect the structure, for example, or alternative translation mechanisms. So of course, we didn't want to stop there though, and we wanted to see how far we can push our method. As we are already doing long read sequencing, we were curious to see actually how long we can go with the reads. And so we next set out to probe the whole HIV genome, well, both in the cell and in the virion. And we were indeed able to do that with 1 reverse transcription reaction and 2 PCRs. And you can see here that the reactivity values are very reproducible with different DMS concentrations, but also with different replicates. Now, interestingly, I think what pops out quite well is a highly structured region right here, which is, of course, the rep response element, but also another region here, which we've heard about before, which is the frameshift element, both showing being highly structured. Yeah, and similarly, we also see this, of course, for the 5' UTR. Now, and I think what is a little bit exciting is that while in previous or older studies, really people had to spend months of work to generate one data set, we are actually able to do this now on a quite low input scale and with very limited work for each sample. So we can scale this up now. And I think there's, speaking of this, we can now try testing out different treatments that we do. And I want to go back a little bit on what we're actually measuring here. So what we think about RNA as folding into the secondary structure, and then we modify it. When we do this native probing, we actually don't have the RNA naked, but it can interact also specifically. It can interact with proteins, and these proteins can change the DMS reactivity. Either it can decrease or increase. And so we thought, why not either treat it natively or first do deproteination and then modify. And indeed, now the changes here then should map to RNA protein interaction sites. And indeed, we tested this first with HV 5' UTR. And while in the cell, we don't see that much difference. If you look in the virion, we see, first, of course, these RNA-RNA interactions, but also we see actually there's quite a lot of positions that change reactivity based on this treatment or not. And so some of these are already known, like the lyse-3 tRNA synthetase, but some of them are novel, and we are currently very interested in working on figuring these out. Now, of course, we were able now to scale this up again, and we were able to apply this now in the whole cell, and the genomic RNA in the cell and in the virion. Now finding multiple putative protein binding site that we're currently working on characterizing further. And as I mentioned before, we're also interested in RNA-RNA interactions. And for these, we can actually just do a different comparison by comparing virion and cell data after deproteination. And we can see the tRNA binding here again, but we also see multiple putative sites that we are going to follow up. And with this, I want to summarize that we get data quality that is quite equivalent to the gold standard with a much simpler workflow. And if you want to learn on this, we have a recent publication, and I want to highlight these tools that are really critical. So with this, I want to thank you for your attention. I'm happy to answer any questions now or later. And I want to highlight Anne-Sophie again for really providing a lot of work too. Thanks.
Great. Thank you very much, Patrick. So let's wait for the questions to come up. But I do have a question. So you have really beautiful work on the virions and the cytoplasm in the viruses. Do you see any difference in the protein bound, unbound regions? Do you think in virion certain regions are more occupied with proteins? Any such information would be interesting.
Yeah, so what I see is that there's definitely a lot stronger signal for protein binding. So I think the most likely conclusion is that there is a lot more protein bound, in the packaged virion compared to the cell.
Great. Another question from Bruno. Thanks, Patrick. Great talk. I would be curious to know what depth of sequencing you need to calculate reactivity.
Yeah, that's a really good, important question. So we did a little bit of subsampling to evaluate that. It really depends on the DMS concentrations. At high DMS concentrations, we need around 1 to 2,000 read or fold coverage, while with lower DMS concentrations, so like 10 or 20 millimolar, like we did for the whole genome, we need around 20,000 fold coverage, which is, for nanopore, then equivalent pretty much to the read number.
Mm-hmm. Awesome. Okay, let's just wait for a few more seconds. In the meantime, perhaps Sahil start sharing his screen. I'll keep the Q&A open, so if there's one more question, we can perhaps take it. Let's thank Patrick again. If more questions come up, Patrick can still continue answering them, so feel free to type them in. With that, thanks, Patrick, again. And next speaker is Sahil Sharma. He'll be talking about endogenous RNA targeting by CRISPR by Cas9. So let's see your screen, Sahil. Awesome. Okay. Yes, works.
Okay, perfect. Hope you can hear me nice and clear.
Yes.
Perfect. So I'd like to first thank Neva for giving me this opportunity to talk here today in this collaborative seminar. We've had excellent talks till now, and so I'd like to switch gears a bit. So from the virus, now I'm going to move you all to the world of bacteria and talk about an exciting and really upcoming field of how CRISPR-Cas systems in the host regulate gene expression of bacteria. So maybe you can get a pointer here. So I would like to start off. So I'm a postdoc working in the lab of Sahil Sharma, and in the lab, we mainly work with two model organisms, an epsilon proteobacteria, Helicobacter pylori, and Campylobacter. So what we're really interested in the lab is looking at how post-transcriptional regulation works in these bacteria. They are pathogenic, they're foodborne. So of course we need cures to prevent them from infecting humans. And the main aim here is, again, looking at how RNA-binding proteins as well and small regulatory RNAs work. Unfortunately, in both of these bacteria, there's no HFQ, so like 50% of bacteria out there. So we don't really know what is a master regulator, if at all there is one in these bacteria, which regulates RNA chaperoning activities and other events. What we do know, and a study performed in the lab some years ago showed us that there are a lot of conserved and strain-specific small RNAs which are present, and the lab is currently busy studying how mechanisms and function of these small RNAs in virulence mainly, and also other stress responses work, and how these control the growth of bacteria. Also, we are also interested in other protein factors, since we lack HFQ, which is a major star in the field of bacterial RNA biology, and also how RNases impact turnover of these RNAs, control mRNA degradation, so on and so forth. But we do have a major player, which you probably all have heard of, is Cas9, and recently Jennifer Doudna and Emmanuel Charpentier were also awarded a Nobel Prize for identifying the basic biology and using it to engineer genetic modifications in various genomes. So this is going to be the star of this talk. And I'm going to introduce to you aspects which probably were unheard of before. So just a brief introduction into what CRISPR-Cas really is. So CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas means CRISPR-associated proteins. And in this figure, we are just trying to briefly go through, we don't have to go into details of this, but as soon as a bacteriophage, which is the virus for bacteria, infects and injects its DNA, if the bacteria has a CRISPR system, it will try to get a memory of this phage infection, which is shown here via a protospacer. This protospacer, part of DNA encoded from the bacteriophage, is then copied into a locus, which is called the CRISPR array, which encompasses all these memories of previous infections. And then next to the CRISPR array are also Cas genes, so CRISPR-associated genes, which really help in all of these adaptation complexes, which really determine which sequences to put in this array. And once they are in the array, then these are transcribed as precursor CRISPR RNAs, which you see here, which is the second stage. And then they are eventually matured into final CRISPR RNAs, which are then loaded onto Cas complexes for, in the case, the bacteriophage, the same bacteriophage infects the cell again, and then you kill them. Of course, it's not as simple as that figure explained. There are a lot of classes and a lot of types and subtypes. So again, you don't have to go into details of what's happening here, but basically these two classes are divided on the number of effector proteins that are present. So in class 1, in different types, you have varied number of effector proteins that are shown here. And in class 2, the major difference is that you have only single effector proteins. So they're not multi-protein complexes, but single proteins working their magic and chopping off phage DNA. And the one that I'm really interested to talk to you about today is Cas9, which is this single effector protein. So majorly, this is broadly the differences between the two classes that you need to keep in mind. You don't need to worry about ones and different. They really basically function all together as one complex to target DNA or RNA. So both of these have the ability, some class types function to target DNA and some function to target RNA. So coming back to our host organism that I briefly introduced was the Campylobacter. As you know, you probably have heard of it or maybe not, but I'm just going to highlight one important fact, that this bacteria harbors the smallest Cas9 that we know in a naturally occurring host. And it's even smaller than the SP Cas9, which probably most of you have routinely used in your lab to make modifications, edits, knockouts, and so on and so forth. But actually, Campylobacter Cas9 is really the smallest. So it's really useful to study this also in terms of delivery methods. If we think of delivering large proteins versus smaller proteins, of course, you would consider the benefits of delivering a smaller protein in here. The interesting fact about Campylobacter CRISPR-Cas system is that it's highly transcribed, and we've always wondered, why are there so many transcripts coming from this CRISPR locus when there's no phage attacking the bacteria? So bacteria is happily growing. There's no phage attack. Why is this CRISPR array producing in large amounts, as you see here in a differential RNA-seq to map transcription start sites? Another important feature of this, which distinguishes this from S. pyogenes, is that in this CRISPR array, there are promoters which are present in the repeats, unlike the SP-Cas9, which has a single repeat, a single promoter in the upstream of the leader. But in this case, we have single promoters upstream of each spacer, which means that every time you incorporate another spacer, you will make another precursor, which is coming from the 5' end to the 3' end of trace RNA, which I will come back to again in a moment. So of course, the question is, when there's no phage, what is this system doing in the bacteria that carries it? Is there a role to it? Is it just transcribing and degrading RNA? Is it just trying to sequester Cas9 so that it does not go and randomly chop off DNA in the cell? So what's going on? And there have been examples in the field where CRISPR-Cas systems and other bacteria have been shown to be important for virulence and other important regulatory aspects. One thing I also really would like to point out to the audience is that while we know a lot about the system in general, about how we can manipulate genomes, what we really don't know is how the systems are regulated in the host, the bacteria that produces these enzymes. Today, I'm going to talk about, while we study all these different aspects of maturation, processing, regulation of CRISPR RNAs, nothing is known how they're degraded. What are the other protein interaction partners of Cas9? So imagine you're working with a Cas9 protein in the host, and we still don't know what other interacting partners of Cas9 are present and what it does at protein level. But I'm going to mainly focus on the RNA binding mode of Cas9. So of course, you've always heard Cas9 as an RNA-guided nuclease and never probably as an RNA binding protein, although I would strongly say that it's also an RNA binding protein first, and only then can it be guided to the DNA. So I will show you some examples of this today. So now zooming in on the system, as I previously mentioned, we have a CRISPR array and then a tracer, which is a transacting CRISPR RNA, which is needed for processing. So you have all these precursors being generated, highly transcribed locus, why are we generating so much that we don't need it because there's no phage attacking? So eventually these spacers and repeats need to be processed into mature forms, and the tracer RNA then hybridizes with the repeat sequence via another complementary sequence called the anti-repeat, which is then processed by RNase 3 into shorter duplexes, and then Cas9 can actually bind this RNP, which now consists of Cas9, CRISPR tracer, and then you can target the phage. But what is it doing in the cell when there's no phage? So to find out what this RNP does, we try to pull it down, we tag the protein natively, and then try to find out what RNAs it binds. Does it bind any RNA at all or nothing? And so this is how we look for novel RNA interactors with Cas9. And we use this technique a lot, and this really works well. So we have identified a lot, which I will now briefly mention to you in a few slides. So if you consider this Cas9 RNP as in simple terms, you will think of three different components, which is Cas9, CRISPR RNA, and tracer RNA. And for it to function perfectly, you need all of this together. So if you remove one or the other, then there's an imbalance there. So we work with 4 different strains of CRISPR in C. jejuni, different isolate strains. So here I'm showing you one strain which has 4 spacers in the CRISPR array. Then there's one other strain called 8421, which has only 2 spacers, completely different spacers because they've been acquired from different phages. And then we also have one strain called GB11, which has no spacer yet. So probably this strain is just waiting to acquire spacer. But again, you would think this strain has Cas9, but it has no spacer. So what is Cas9 doing with the strain? And I like to visualize this very simply in terms of how the complexes look in 4 different strains. So you have this complex here, which is fully occupied, 4 RNAs, highly transcribed. And here you have a complex of only 2 spacers. So is there Cas9, which is freely available, ApoCas9? And then in this strain, which is the last, which has not acquired any spacer yet, how do you process tracer? What is Cas9 doing? Is it just waiting for tracer RNA spacers to be acquired? So what's happening in there? And of course, as you would probably imagine, each of these comes with a promoter. So the more number of spacers you get, the more promoters you get, and the more highly transcribed the region is, the more competition to be loaded on Cas9. So the more spacers means, the more abundant Cas9 RNP, and it changes with that. So again, the question that we're trying to address is, without looking at phage and defense, is does the system has other functions beyond defense? So what's it doing in the host? So one of the early clues came from a study published 2018, where this Cas9 RNP that I just showed you, which has a CRISPR and a tracer RNA and is basically waiting for the phage to attack with this guide, actually recognizes complementary sequences in some cellular RNAs. So this leads to hybridization of this, and then some of these RNAs are cleaved. So this leads to endogenous RNA cleavage, and this has also been shown for SpCas9 and Staph aureus Cas9 by other groups at the same time. The other important aspect that we recently discovered was how tracer RNA actually impacts cellular RNA. So I mentioned there's a tracer RNA in this GB11 strain. So you can imagine it does not have a spacer yet to hybridize to. So what it does, it basically finds imperfect partners to pair with. So cellular RNAs at the 5' end of tracer RNA, and then you find complementary sequences. Then Cas9 thinks that, oh, this is the perfect spacer for me, I can cleave it. And then this leads to generation of non-canonical CRISPR RNAs. And this was the first example of CRISPR RNAs being generated, not from the CRISPR array, but from a different location in the host. So this also led us to develop a multiplexable diagnostic platform, which you can further read in this paper. The third very interesting aspect that I'm going to show you briefly today is about CRISPR-independent targeting. So what happens in strains where you have Cas9, but you don't have tracer or CRISPR RNAs yet? So there are other RNAs in cell. Cas9 does recognize double-stranded RNAs. So is it a possibility that it's going to target these? And how do you study this in respect of independence of CRISPR system? One thing before I go ahead, I would like to point out is that while we all know Cas9s as these guided nucleases, but they actually are very specific in terms of how they recognize RNA structures. So for Cas9, as I mentioned, a tracer RNA, this has a very peculiar triple helix at its 3' end. So the Cas9 will not recognize any tracer RNA from ortholog Cas9. It needs its own tracer RNA. Similarly, the S. aureus Cas9 also has a different stem loop at its end, which is completely different from the stem loop found at the SP Cas9, and so is the same case with Francisella Cas9. So all of these Cas9s do have bilobe structures, bind, and are guided to DNA, but in terms of recognition of these tracer RNAs, they're really, really specific, and you cannot change the 3' end of these tracer RNAs. So while being, very similar in targeting, they're very different in how they recognize some of these 3' ends of tracer RNA. So this has to be kept in mind before I go really. So what we decided was, let's just make some strains where we remove the CRISPR system and also add a complementation strain where we can then add the dCas9 complementation, so that to see if we can catch some targets, which we usually miss because Cas9 is an active protein. It will clear as soon as it binds. So we generated some strains which like either the CRISPR or the tracer or all the CRISPR RNAs or all CRISPR tracer RNAs. And you can clearly see the impact that it has on regular levels of these. So in the wild type, you clearly see a lot of tracer CRISPR RNA present. As soon as you remove the tracer RNA, because now it cannot process CRISPR RNAs, we lose all of it. Then you remove CRISPR RNA3, it has a slight effect on tracer reduction. You remove the whole locus, of course, we cannot see any of these. And then you remove the 4 CRISPR RNAs, the abundance of tracer goes down because, of course, now you remove the 4 promoters. The impact on protein is not so much. You can clearly see that Cas9, which is flag tagged in all of the strains, work similarly. So we don't see a reduction of the protein level. So we're not hampering Cas9 protein by removing any of these. The other aspect, as I mentioned, was the Cas9 and the dCas9 system, where we complement a deletion strain with either the wild type and mutant. And we can also clearly see that the mutant dCas9 does not impact the ability to bind CRISPR RNAs compared to the wild type. So it's perfectly functional. When we looked at the RNA targets, we were really surprised because then the targets of these Cas9 that were bound exponentially sort of increased as soon as we went on decreasing, tracer, then further increased to the CRISPR, and then as soon as you delete the whole locus, you have these bunch of RNA which are bound. So what happens that now suddenly Cas9 is binding so many RNAs in the cell? Most likely because it was occupied by spacers previously. The same happened with dCas9. So dCas9 now does not have the ability to really cleave RNAs. So now we really uncover those RNAs which were initially lost by precipitation because they were cleaved by an active enzyme. I will now really focus on one locus here to show you just a brief of what's going on right now and how we identify different targets in this. So again, this is a slide showing a snapshot of a locus called the CME-ABC. And here you can see wild type and different deletion strains of Cas9, of CJH9 and we pull down with Cas9 flag, and you can see differences happening as soon as you remove one or the other. In the delta tracer CRISPR and delta CRISPR tracer strain, we see we are now basically binding this CJ0369 locus, which initially was not able to be detected. What this locus is important for is just downstream of this is a CME-ABC pump, which is an important multi-drug efflux pump. So this CMER is a regulator of this pump. So by regulating the upstream gene in this operon, we think that Cas9 might have a role to play. Of course, I know what the other question that many of you in the audience are thinking is, oh, this is an artificial system where you didn't really tracer CRISPR.
We might have a technical problem here. Let's just wait. You can, in the meantime, perhaps type your questions already. Sahil, it stopped. But anyways, we are running out of time. So could you start sharing again and perhaps wrap it up, please? And we take the questions. Thank you.
Yeah, this was the last slide. Yeah, so yeah, basically with the different methodology, we're trying to get what Cas9 is binding in the host, irrespective of the CRISPR and tracer-dependent mechanisms. And we think that this is an important thing to study because we've developed technologies based on these, and we should know how Cas9 actually works in the host from where we actually take it from. And yeah, this is just basically what is known, and we're looking for what additional roles it could have. And with this, I would like to thank the lab, Cynthia mostly, and also other collaborators from the Beisel lab, Chunlei, and other members from the core unit. And sorry for being over time, but I'm looking forward if you have any questions. Thank you.
Thank you so much, Sahil, for this beautiful talk. We are a little bit over time for the question and answers, but yeah, let's just wait for a moment to get the questions. My question, I can perhaps start with one question. So you showed that interesting gene that was showing the difference in the effluxor, right? There was a pump. Why do you think specifically you see the difference there in the CRISPR and tracer mutant? Why this is?
So I think when you delete these RNAs, you of course free up the available Cas9, because now Cas9, which was initially blocked or has the preferred substrate, which is a CRISPR or a tracer RNA, is now free. But like we saw in this other strain where we don't have a lot of spacers, but still, Cas9 is able to bind the same locus. So I think it's just a combination of the amount of the preferred substrate versus, or under conditions of, different environmental stresses where this could be adapted, I think.
Mm-hmm.
Yeah.
And the binding seems also the structural changes of the Cas9 upon tracer binding seems to also change its preference for RNA targeting.
That is a possibility. We don't have structures with just tracer RNA, so we cannot really say much, but yeah, possibility exists. And as I mentioned, the 3' end of the tracer is a triple helix, which is highly structured. So if these RNAs that are already being bound by Cas9 are structured, that could probably also be a hint. Yeah.
Thank you. Let's wait for other questions from the audience. If that's not the case, I think we can then conclude. The next RNA Collaborative Center seminar will be on October 25th by Harvard Medical School. So please register if you haven't done so. It was a pleasure hosting the RNA Collaborative Seminar from Würzburg, and wish you all a great day. There are some questions coming up. Perhaps, Sahil, you can answer them. Hang in here. And yeah, we thank the rest, and I would like to conclude the session here. Thank you.